Hi, everyone. Welcome to the third session of the symposium today. My name is Noah Mardix Rappaport, and I'm a senior research scientist here at ISB. I'm very pleased to open our next session of the symposium, which will focus on the functional consequences of evolution in host microbe systems. So essentially how the evolution of the microbiome affects health of the host. In this session, we will have two speakers, Dr. Tami Lieberman from MIT and Dr. Taichi Suzuki from Max Planck Institute of Developmental Biology. As in the other two sections, time for Q&A will be right after the two talks. So please make sure to deposit your questions in the Zoom using the Q&A button at the bottom of the screen. And please don't forget to note which of the speakers the question is intended for. You can also upvote other questions. Each talk will be 30 minutes. And after both talks, we have about 30 minutes for questions to both speakers. Tami, Dr. Lieberman, sorry, this is your cue to start sharing your screen. It is my pleasure to introduce our first speaker, Dr. Tami Lieberman. Dr. Lieberman is an assistant professor in the Department of Civil Environmental Engineering and in the Institute of Medical Engineering and Science at the MIT. Tami developed genomic approaches to study how bacteria evolve either during infection or as they colonize humans, and also developed new platforms to visualize bacterial evolution in real time. Her lab does both computational and experimental research, focusing on uncovering the principles governing colonization, niche range, and personalization in the human microbiome. And she will talk to us today about tracking de novo mutations to understand microbial ecology on the skin. Take it from here, Tami.
Thank you so much, Noah, for that introduction. Thank you so much, organizers, for having me here. I'm giving this talk today from a hotel room because I was actually planning to be traveling. I am traveling for a friend's wedding, but I couldn't really think of a better way to spend this morning than listening to these great talks on topics that's so interesting to me. Okay, so what this talk today is about is mutations in the microbiome, and really not so much the population genetics of strains, but what are the mutations, de novo mutations that are happening in each person, and how can we use those mutations to better treat diseases and engineer probiotics. And we're excited about this area because I believe in the power of probiotics for one day really treating a whole host of human diseases and promoting wellness. But there's a real problem that's sort of inhibiting us reaching this future potential, which is how can we get these microbes to really stick in gut microbiomes long-term, or skin microbiomes or any microbiomes long-term. And this problem is illustrated by this nice paper here, which is the best example I've seen so far of getting a microbe to really stick in a microbiome. And what we can see is that after removal of probiotic administration, we can't detect this probiotic in half the subjects. And so we don't know why this microbe had some chance of sticking in some subjects, and what really is distinguishing these different groups of people. And the problem with sort of really engineering these microbiomes in sort of this precise way is that we're still really in the early days of the microbiome field. There's still just a lot we don't know, right? We don't know what the niche range is for an individual bacterial strain. Now, which bacteria are the bacteria that are currently living on me? Do they have any potential for living on you? We don't know what the selective forces are that determine these different niche ranges. And we don't know what the role of within-person evolution is, right? Maybe the microbes that are already living within me are adapted to some unique set of selective forces that I have, or my microbiome has. And so these new bacteria from the environment aren't going to stand a chance against these pre-adapted bacteria. What's exciting about tracking within-person evolution is that not only can we learn about within-person evolution as a phenomenon on its own, but we can use it as a tool to address these other questions as well. And what I mean by that is that we can by tracking within person evolution, we can reconstruct transmission network, both between people and within an individual person. And by looking at what genes are driving adaptive evolution, we can understand what genes are most critical to bacterial survival in vivo and infer selective challenges. And so given this potential of evolution as a tool, and given that really just simple back of the envelope calculations reveal that there's got to be a lot of potential for evolution and adaptation in our microbiomes every time our microbiome replicates, which is probably once per day, you might be asking yourself a question, which is, why hasn't within-person microbiome evolution been studied more? And, I still keep this question here, despite the great papers that were mentioned today, the great work that's been done, because all those papers came out, just in the past couple of years, right? And this microbiome field's been going on for a long time, and metagenomics has been available for a long time. So why has this sort of been maybe underappreciated? And there are many possible reasons, but I'll give you a couple. One is that I'm like a little worried that my setup is not good, and so please tell me if you can't hear me. But okay. Anyway, so why isn't this studied more? So one is a conceptual misconception, which is that when people think of studying the evolution, that we think often that we need to collect time series, and maybe even long time series, to be able to watch some weak selective pressures in human guts. And no one really wants to do a PhD that's quite that long. And so getting such data is difficult. But in reality, while this longitudinal data is really important and really powerful, we can often learn a lot about evolution by just looking at the coexisting diversity within a single person at a single time point. And we can do this because what we've learned from my work and from others is that as bacteria are adapting within individuals, even if they're adapting under pretty strong selective pressures, often these adaptive mutations don't take over in the population, and they can coexist for some period of time with other adaptive mutations. And so this coexistence allows us to use evolutionary inference from just a snapshot at a single time point and project back into the past. Okay, so we have this technical problem, but there's this conceptual problem, but there's also a technical problem, which is that, of course, 16S doesn't provide lineage level resolution. And so we need to use metagenomics if we want to sort of use culture independent approaches. But metagenomics is really difficult to use for understanding within person evolution at sort of this mechanistic level. And this is something that's been kind of alluded to before, but I want to mention it again. Because what we can see is that people can be colonized by two different strains, or what we call lineages. And these lineages can bring with them coexisting variants that come not from a mutation that happened to that person, but from some mutation that happened sometime in the past. And we want to be able to distinguish these pre-existing variants from de novo mutations, right? Mutations that are actually happening within a person. And perhaps we can write some cool techniques, or we can use some of the techniques that Ben and Andita talked about. So sort of remove these variants that are coming from pre-existing variation and just get these de novo mutations. But even if we just have these de novo mutations, getting to great interpretation can be hard because we often can't phase mutations or figure out which mutations are in the same cell. And this is a really hard computational problem. To get a sense, this is just a phylogeny of isolates from one long-term infection. And you can just see how complicated it is and how hard that would be to reconstruct from some cool minimum strain algorithm. And so what we do is instead we culture for single colony isolates. And because each colony is formed by a single cell, we can sort of, when we sequence their genomes, we can infer, we're really inferring the genomes of single cells that existed in that person. We then sequence their whole genomes and focus on the mutations that just happened on the tips of our inferred phylogeny. And this, Ben sort of alluded to this, but I want to mention it again, which is that not only can we get the linkage using this, this technique, but we can call mutations of the entire genome, which can really get us rates of mutation and help us understand and interpret these things. In addition, what we can do is really identify these variants that are low frequency, really these transient competing alleles, which let us identify parallel evolution and adaptive signals. For those who are interested, we use an alignment-based approach, but we also do de novo assembly first. So we first use some sort of rough alignment to cluster isolates into closely related lineages, build lineage pan assemblies, and then use those new assemblies to get really clean alignment-based phylogenies. And so using this approach during my postdoc, I work with graduate student Jay Zhao, and this work has been mentioned a couple times today, to study the evolution of Bacteroides fragilis, a common gut bug, in 12 healthy subjects. And we identified many cases of within patient parallel evolution. And these genes that were mutated multiple times also showed elevated DNDSs, showing that they really are under adaptive evolution, while genes that did not show parallel evolution had a signature of neutrality. This pointed to key genes that were involved in within-person evolution, including these importers of complex polysaccharides and these genes that are important for how the bacteria look on the outside. And one thing that was really cool from this study and allowed us to get was to get a really nice fine-scaled observation. I'm very sorry about the background noise. We're just going to have to hope that my voice is louder than it. So from this study, we observed this really interesting phenomenon where from colony-based sequencing, we were able to make a really clean phylogeny, but follow this phylogeny using metagenomic sampling taken every other day. And combining this, we built these diagrams that show how we infer evolution to have occurred in the subject. And we infer is that prior to sampling, about maybe 8 years prior to sampling, there was a single-celled ancestor that diverged into two different coexisting lineages. And these two different coexisting lineages continued to coexist during our whole 2-year time series, despite the fact that there were really crazy dynamics in the top one, where this blue genotype emerges and outcompetes its ancestor without influencing the coexistence with this orange genotype. And so this suggests something really interesting, right? That maybe these bacteria are functionally diversified and they're doing different things. But there's a real problem in the gut microbiome, which is that we can't distinguish this from just separation in space, right? Maybe these lineages are just coexisting because they never see each other. And so in my lab, we've been focusing more on the skin microbiome, where we can ask questions directly about space. And we've been doing this. And so the first story I want to tell you is led by postdoc in my lab, Erilyn Conwell, and it asks, why are strains coexisting in the skin microbiome? And we can imagine two different alternative hypotheses, sort of adaptive explanations like niche partitioning, or sort of neutral forces like separation in space. And to address this question, we decided to focus on the sebaceous microbiome, which is the microbiome of our face, backs, and chests, because it's easy to sample at many spatial scales. It's relatively low diversity, so we can maybe understand all the individual species and how they behave as a community. And it's also stable over time, so it's not just some sampling from the environment. And when I say it's low diversity, it's a really low diversity. There's one bacteria, Cutibacterium acnes, that's dominating the majority of the community. But it's not that boring, because there's actually quite a lot of strain level diversity within Cutibacterium acnes, and the reasons for the strain diversity are not well understood. C. acnes is called acnes, and so you might think that it's a bad guy, but we're going to consider it a commensal because it's literally on everybody. So to study C. acnes, we took samples from healthy subjects using a toothpick to get these sort of coarse, broad geographic samples, and also single pore samples from just comedone extractors, which is a cosmetic tool. It's kind of like squeezing a blackhead out of your face, and pore strips that helped us get some better spatial resolution. From each of these samples, we then cultured many single colonies and performed whole genome sequencing. The first thing we can see from this kind of data is that we can recapitulate the idea that there are many different coexisting strains on each person. To get an example, here, this is just one subject across the whole tree of C. acnes. And here, the scale bar is very large. And we can see that on this whole tree of C. acnes, subject A has lineages from all over this tree. And particularly, even coexisting lineages that are relatively close to each other in terms of the global phylogeny. And this is something we see on all the other subjects as well, where there's many strains on each person. And when we go and we look at where these strains are separated in space, we don't see any strong signal for spatial location where there are strains that are maybe only on one area or another. And we don't really see any strong signal for which bacteria we get from the surface and which ones we get from the pores. Okay, so they seem to be maybe everywhere, but we do see something that's not sort of cosmopolitan when we look at the level of individual pores. When we look at the level of individual pores, we can just think about the statistics of picking 2 colonies at random from that patient's population. And if we were to run these simulations, what we find is that, we'd expect pairs of pores from the same person to have the same strain about 50% of the time. And of course, this is different for each person, depending on their distribution of strains. But from this person, what we see is that really almost all of the time, we get the pores from the same, colonies from the same pore are almost always from the same lineage. And we see very similar trends on other subjects as well. So there's these strains are everywhere, but within a pore, there's only one strain. Or there's one strain that's really dominant, right? So just suggesting that each strain is sort of dominating its own island. And we can even zoom on this even further. And here's just an example of a pore strip from a different subject, where we see 3 different pores that are within 10 millimeters of each other, and each one is dominated by a different lineage. And so this might suggest that maybe each pore is filtering for a different type of lineage. And to understand this better, we can zoom in and look at the diversity that's happening within the lineage, and sort of and look at these de novo mutations that happened on that person. And when we zoom in to this fine detail, we see something really striking, which is not only does each pore have one lineage, each pore seems to have one really closely related genotype, where we find, mutations that are private to a pore, at least to the amount of sampling that we've done on these subjects. So there's really relatively low diversity within each pore, despite the fact that there's lots of diversity on that subject, suggesting that it's not filtering at the level of lineage, right? If there was filtering at the level of lineages, you might expect, multiple colonizations of that pore from different genotypes within that lineage. And this, again, is something we see across subjects. And so what's going on is maybe there's not just filtering at the level of lineage, but there's actually selection that's happening for individual genotypes for each pore. And if this were happening, we should see strong signatures of adaptive evolution. But we don't. We see really everything looks super purifying, where we see a depletion of non-synonymous mutations no matter how we cut it, and we've cut it lots of different ways. So there's not there doesn't seem to be if adaptation is not driving these bottlenecks, then what's going on? What else could be causing it? Well, it's not something random, right? Because these pores, it's not just like a simple trivial explanation, because these pores really do have millions of bacteria in them when we sample them. And what we think is going on is that there's a bottlenecking during the earliest colonization of these pores, where when pores are opening up for colonization by some mechanism or another, it's actually pretty hard for a bacteria to get to the bottom of the pore, because the bacteria aren't growing very fast at the top of the pore because it's aerobic, and they really prefer to grow in anaerobic conditions. And the bottom of the pore is also where the food is being secreted, the oil and sebum that we're secreting is being provided to the bacteria. And so once these bacteria are able to fight sebum flow and get into this preferred zone of growth, they can quickly grow and expand before the next rare migration event can happen. And there are other possible explanations. This is just one that we're thinking about right now. But what it means is that we think is going on is that there's some sort of priority effect arising from physiology. And we think this can explain coexistence of strains. Why? Because we know that when you grow these strains in the lab, they really do have different fitnesses, at least in the trivial conditions that we've put up in the lab. And so something's driving them to coexist. And we know that the growth on the surface is relatively low, and most of the growth is happening in these pores. And these pores, they're not competing with other genotypes, right? And so even if this purple genotype can't really compete with the green genotype, it never really has to. So what I've shown so far is that we think that C. acnes lineages are spatially segregated in different pores, that adaptive evolution is almost negligible in these scenarios, in contrast to things we've seen in other systems. And we think that these bottlenecks that are happening at the level of individual pores are contributing to this coexistence and maybe even this lack of adaptation. And so there's a section about implications for disease, and I do think that these results have implications for therapy. Because when we see strong adaptive evolution, like in Bacteroides fragilis, we think we might need to pick, and what this tells us is that each person may have different selective forces happening within them. And so maybe we should pick the bacteria from our collection, the probiotic from our collection, that's best able to compete within the selective environment. But in the case of the skin microbiome, fitness or genotype doesn't seem to matter that much to coexistence or in survival. And so for these pore dwelling species, perhaps we don't really need to worry about competitive ability, and instead we should focus on removing resident strains. Okay, so with the last section of my talk, I'll quickly tell you another story where we think adaptive evolution may be playing a bigger role in disease. And so instead we're going to be focusing on disease as a bacteria that's involved in disease rather than health. And it's a bacteria called Staphylococcus aureus, Staph aureus, which is known for a long time to colonize the lesions of people with atopic dermatitis or eczema. Eczema is a very common skin disease. It causes the itchy patches of skins. It's very common in children, up to 20% of children. And it's known, again, that staph colonize these lesions. And for a long time, people have sort of associated in some mouse models that the strains that are isolated from people with AD maybe have more disease-causing debility than strains that are isolated from healthy people. So we conducted a longitudinal study of children with AD, classic severe moderate symptoms, conducting longitudinal sampling and our culturing-based approach. The first thing to know about this disease, first thing to know about the population genetics here is that each patient is colonized by a different strain. There's not some AD unique strain. Really, the strains that we're isolating span the diversity of Staph aureus. And for some reason or another, each person has one strain that's stably colonizing them for a long time. So we can ask within this strain, what's happening at the mutation level. So here's one phylogeny from one subject, and other subjects have very similar patterns. So I'm just showing you one here. And I've colored this phylogeny by where isolates were taken from. And we can see that really there's no segregation over space, right? That isolates seem to be widespread across these different locations. But when we look at a time signal, we do see some segregation here, where these genotypes at the top were taken later. And this may be hard for some people to interpret, so we make these diagrams over here, which show very nicely that new genotypes are emerging over time and sweeping the population. So we really are seeing adaptive evolution happening, or not, sorry, we're not seeing adaptive evolution, but we're seeing body-wide sweeps happening in these individuals. And so this evolution could be, and we're seeing this in other subjects. And so evolution could be adaptive, or maybe these patients are just washing themselves really well and some lucky genotype is taking over. And what we see is that there's actually quite a strong signature for adaptive evolution. And in particular, we see one gene where there's a really strong signature of parallel evolution, where in the same tree I just showed you before, we see 4 mutations in the same gene, even though there's less than 100 mutations on this whole tree. And we see a similar gene have another sort of loss of function mutation. And this gene is called CapD. It's involved in capsule synthesis. And we see it mutated, not just mutated 4 times in the subject, but one of these mutations is involved in the sweep that we see. And we see a different mutation in a different subject also involved in a sweep. Okay, so what is this gene? This gene is essential for capsule production. And when you get loss of function mutations in this gene, you become capsule negative. You lack the sugar coating on the outside. And that seems super weird, right? Because capsule is generally thought to be a good thing that protects you from the immune system, which should be active in atopic dermatitis. Capsule is known to be critical for survival in a lot of different mouse models. But for other bacterial systems, capsule variation has been shown, and maybe even shown to have some body site specificity. So maybe it's not super surprising, but the mechanisms of sort of this trade-off is not well understood. Another interesting thing about this is that an epidemic clone of Staph aureus that causes not just Staph aureus, but methicillin-resistant Staph aureus, USA 300, also lacks a functional CAP-D. So, and it's really involved in skin diseases that are acquired in the community, suggesting that maybe capsule loss helps on skin. And so we wanted to see if this might be true with a mouse model in collaboration with Isaac Chu and Jean Lee. And really these experiments were done by Veda Kadka in my lab, but we're new at mouse models, so we needed some help. And what we found using an isogenic set of lab strains, or what we thought was an isogenic set of lab strains, was that these CAP-D mutants do cause worse disease in this epicutaneous model. I do want to caution this though. We did this experiment using these isogenic strains, or what we were told were, these deletion strains in this lab strain that were provided to us and that had been existed for a long period of time. And we're not completely sure now that we know that these strains are not completely isogenic, that there is another SNP in the CAP-D mutant relative to the wild type that's not at this locus. And so we're repeating these strains with these clinical isolates that we were finally able to get IRB approval to work with and be sent out of our collaborators lab in Mexico. So please hold on to the story, but tentatively we think that CAP-D deletions cause worse disease in mice. Regardless of whether or not they cause worse disease, we know that this is a common thing and not just special to something in our study, because when Felix, the first author in the study, downloaded public data and reanalyzed that to see whether or not they had the full-length CAP-D gene, we find that when you take isolates from people with AD, a large percentage of them already have this CAP-D truncation or a loss of function, either from de novo mutation or maybe being colonized by a CAP-D gene, CAP-D minus strain. But isolates from healthy people almost always have the full-length gene, and as well from other infections. So what we're seeing here is that even though there's not really an association between, or a difference between disease and health in which strain of Staph aureus you get, at the level of this individual gene and whether or not you have this gene, there does seem to be a difference between health and disease. So we see, so here is that Staph aureus lineages rapidly spread across the body in people with AD. And one of the things that's happening here often is CAP-D loss. And what's exciting for me, to me about this in terms of therapy, regardless of whether or not these mouse models replicate, is that we know that these bacteria really are adapting to sort of survive better on the skin. And so if we want to clear Staph aureus from the skin of people with atopic dermatitis, we need to make sure that our probiotic strains are active against these phenotypes that often emerge on people with AD. And so we need to make sure our probiotic strains are active against capsule negative strains. Going forward, I've shown you a case where adaptive evolution dominates and a case where neutral evolution dominates. And we have some ideas for maybe why that things are different here, but we need to figure out what the broader rules are for determining whether within-person evolution is adaptive or neutral. When we see these things that look like they might be loss of functions in key genes, it'd be important to understand if these are long-term dead ends that aren't going to spread to another new person, or if these are really adaptive strains that are emerging that might be successful across individuals. And of course, just as everyone else here is interested in, we're interested in the role of on-person adaptation to both ecology and disease. Okay, so I mentioned the main collaborators. I think I kind of went over it fast. So I'll mention here that this Staph aureus work was really driven by a postdoc in my lab, Felix Key, who's now a group leader at the Max Planck in Berlin. And was really a critical person to this work was Maria Teresa Garcia Romero, a dermatologist in the National Institute of Pediatrics in Mexico City. So with that, I'm looking forward to the next talk and taking your questions later. Thank you.
Thank you, Tami, for this fascinating talk.